Very good evening, children. We'll start our today's class. Today's topic is about animal cloning. We're going to talk about animal cloning. So first, let me define what is a clone, and then I'll explain you about how the animal cloning was done. Cloning is actually a process by which morphologically similar, genetically similar organisms are produced. It's a process by which morphologically, that means structure wise, genetically, that is the gene wise, we can produce similar organism. That is called as cloning. Cloning is one way of asexual reproductive methods. Cloning, sometimes it occurs naturally, sometimes it has to be induced, or it can be artificial also. And here cloning, why it is referred in biotechnology, here we are creating copies of organisms, similar organisms, by using the reproductive cells. That's why cloning, it is there under the biotechnological process. Now let us see about the list of cloned animals so far. In the year 1997, in Scotland, the first cloned sheep dolly was done by Dr. Ian Wilmot. In the year 1997. In the year 1998, an international team of scientists, they have announced that they have cloned 50 mice. And also in the year 1998, the same year, a pair of cloned calves were born in Japan. And similarly, in 1999, researchers from Massachusetts, they have identified and they have produced the birth of 3 cloned goats. They have identified, they have produced. And similarly, in the year 2000, 5 cloned piglets were born in United States. And in Texas, the first cloned cat, it was born in the year 2001. 2002, French scientists, they have announced that they have cloned a rabbit. And the year 2003, a mule, the first cloned mule, it was born in Idaho. And similarly, in the year 2003, the first cloned horse, it was born in Italy. The first cloned organisms, this was done by Dr. Ian Wilmot, are the first cloned mammal, that is called as sheep. It was named as dolly. Now let me explain you how they have developed this particular cloned sheep. And also I have mentioned clearly that the cloned sheep are asexual organisms, asexually method of producing organisms. Here the sexes are not involved, but the fusion of gamete takes place. How this process takes place here? For this experiment, Dr. Ian Wilmot, he has taken 2 different types of sheep. One is a 6-year-old Pin Dorset sheep. This Pin Dorset sheep he has selected, and similarly he has selected a Scottish blackface, and in both the cases he has isolated the cells. The cell he has isolated. Here from the Pin Dorset sheep, he has taken the cell from the udder cell. Udder cell means from the mammary glands, he has taken the cells. So he has isolated the cell from a 6-year-old Pin Dorset sheep from the mammary gland or udder cell. From that he has taken the nucleus. He isolated nucleus. Similarly, From the Scottish blackface, he has taken the egg cell from the ovary. From that egg cell, he has removed the nucleus. Now we can see that there is an egg from Scottish blackface without nucleus, and here other cell he has taken and has separated the nucleus. Then what happened? The nucleus of 6-year-old Finn Dorset sheep and the ovum of Scottish blackface, both of them he has fused together. So the nucleus, it is from the mammary cell of the Finn Dorset sheep, whereas the ovum is from the Scottish blackface. Now this culture, this egg with a foreign nuclei, it was cultured, it was grown in a separate culture medium, and it starts dividing and it developed into an embryonic stage, an 8-cell stage, a blastocyst stage. In that stage, he has implanted that embryo into the third sheep, that is the surrogate mother. In that uterus of the surrogate mother, the whole development of the baby took place, and finally it gave birth to a sheep. That sheep was exactly genetically, morphologically, it is similar to the 6-year-old Finn Dorset sheep. 6-year-old Finn Dorset sheep. So this indicates that from where we are taking the nucleus, that play an important role, because the nucleus is the one which controls the morphological characteristics as well as genetical characteristics, because nucleus is the one which carries the genetic information from one generation to the next generation. So the nucleus, it is taken exactly from the Finn Dorset type of sheep. So even the lamb Dolly, it looks like exactly the Finn Dorset sheep. This is how the clone animal, the first sheep, it was cloned. Dolly was cloned. Now we are going to talk about the advantages and disadvantages of cloning. Cloning, it has its own benefit and disadvantage. First thing is this advantages of cloning. It offers benefit for various clinical trials and medical research. Lot of animals we can clone it and we can use that for research purpose or clinical trial to check some vaccines, like that we can do it. And also it can be useful for the production of proteins and drugs in the field of medicine, or to design a drug for in the field of medicines. And also it plays an important role in stem cell research. And through cloning, animal cloning, we can save the endangered organism or endangered species. Similarly, this cloning, the disadvantages are a big threat to the biodiversity. Because of cloning, if you are going to end up in a different characteristic organisms, it alters the whole evolutionary process and it will have a very big impact in the population of the ecosystem. The next thing is this process is very, very tedious and more expensive. It requires a lot of technique as well as technology. It can even cause animal to suffer, because you are going to extract the nucleus, extract the ovum, cells. And also the various reports of cloning, it clearly tells that the surrogate mothers were manifesting adverse outcome, and the animals that are cloned were more prone to diseases, and then the mortality rate, the death rate, it is very, very high when compared to naturally developing populations. If the cloned animals, if they are going to be taken as food for the other animals or human beings, it might causes lot of ill effect in their body. And also the clone animals, it attains the age far faster than the normal animals. And these animals, when you noted down, they're all less healthy when compared to the parent organisms. They're not more stability. And also cloning can lead to occurrence of genetic disorders in animals. These are all the various advantages and disadvantages of cloning. Next, we are going to talk about the next topic is about branches of biotechnology. The various branches of biotechnology. The branches of biotechnology. We have red biotechnology, green biotechnology, white biotechnology, and blue biotechnology. These are all the four different fields or branches of biotechnology. This red biotechnology, that means it mainly involved in the production of antibiotics, vaccine development, genetic engineering, cloning. All these genetically related studies, it will come under red biotechnology. It actually deals with improvement in medical drugs, improvement in medical technology. These are all the red biotechnology. The next technology is called green biotechnology, deals with agricultural processes, development of latest equipment, technological equipment in biotechnology, production of new variety of crops, new variety of products, genetically engineered organisms, genetically modified plants, production of food quality, resistant varieties, drought resistant, heat resistance, temperature resistant varieties, pest resistant, like that, different types of production of different types of organisms, different types of genetically modified organisms. It comes under the green biotechnology. It deals with agricultural processes. The third is called white biotechnology. It deals with industrial products, production of use of biotechnological tools for production of chemicals and energy. It is also called as industrial biotechnology, production of energies, chemicals, various polymers, paper industry, food industry, processing industry, production of enzymes. These are all, it comes under the white biotechnology, including the production of the various beverages, biodiesels, everything, it comes under white biotechnology. And the fourth category is called blue biotechnology. It deals with application of molecular biology, the field of marine, water, aquatic microorganisms. Usage of microorganisms or usage of agricultural, usage of the aquatic organisms for medicines, for production of various pharmaceutical products, for food as well as the feed supplements, like using the fish meal to feed poultry, like that. When you go discuss with that, that is comes under the blue biotechnology. So red biotechnology deals with the medicines, antibiotic, vaccine, gene therapy, parasite, the pharmaceuticals. Green biotechnology, agricultural, environmental protection, and food biotechnology. White biotechnology, food, feed, energy, chemicals, polymers, and pharmaceuticals. Whereas blue biotechnology, it deals with medicines, food, and feed. Next topic, what we are going to discuss today, it is about biosafety. So next topic is about biosafety. So what is this biosafety? When you are working in the field of biotechnology, we are handling so much of microorganisms. Some microorganisms, it may be pathogens, it can cause diseases, or some products obtained from those microorganisms, it can become a toxic to the environment as well as toxic to us. It can be an harmful biological agent. It may even cause the extreme death. So what are the procedures or what are the policies and what are the principles that we have to follow while handling these type of organisms in the lab, in the place of work, to safeguard the environment and the human population and the people those are working there. That is called as biosafety measures. This biosafety is not only for the people those are working there, but also to ensure it is good for the public and it is not harmful to the public, as well as it should be environmentally safety. That is the ultimate aim of biosafety. This biosafety, there are certain protocols that we need to follow. That means the people should have made aware of the rules, the safety rules, the regulations, and who is the monitoring body which controls that. For example, pollution means pollution control board, which is where it is located, and what is the protocol given by them. All those should be, people should aware of that. And similarly, researchers, the people those who are involved in those research must know the complete knowledge about the biosafety issues, or they should know the way to protect themselves when there is a problem at any level in that. And also, this biosafety rule, it is not only for pertaining to only one country, it is throughout the world. And there was a multilateral agreement about the Cartagena Protocol on biosafety. This was adopted by 167 countries, including the countries of United Nations, to exchange their ideas, to exchange about the knowledge about the toxic substances or organisms. And this protocol was enforced from 11 September 2003. What are the objectives in which the protocol was enforced? Mainly to ensure the safe movement of living modified organisms across the boundary. When something has to be transferred from one country to next country, what are the safety measures they have to follow? Similarly, to frame a regulatory rules, a regulations, and share those principles and methodologies to protect or to safeguard the people around that. So that framing the laws and sharing those principles and methodologies through biosafety clearinghouse. These are the two important objectives of the Cartagena Protocol on biosafety. Importance of biosafety issues. So how important these biosafety issues are there? The place where the research or genetic engineering practices are done, they should get a prior approval from the regulatory authorities of the country, and they have to get the guidelines from the authority and follow the guidelines to minimize the problem that they're going to face there, the biosafety measures. And also the awareness about the biosafety, it should be there for the people those are working there, the researchers, the producers of genetically modified organisms, the policy makers or lawmakers, the administrators, and the environmentalists should know about what is going to happen there. There is a company that is called Omics. It is a publishing group in USA. Every year they'll publish a biosafety journal, and also they'll organize international conferences on biosafety to make everyone aware of the biosafety rules, what is framed in different countries, and how to handle those organisms. And also the biosecurity and bioterrorism. This is a very emerging issues nowadays. What is bioterrorism or biosecurity? The bioterrorism means they're using biological organism to kill human beings. That's called bioterrorism. This need to be checked upon in the interest of humans and environmental safety. And there are certain biosafety levels are there, level 1, level 2, level 3 and level 4, based upon the laboratories, based upon what type of organism they are going to handle, what type of pathogenetic organism they are going to handle. And this levels, it varies. It depends on the environment and depend on what type of organism they are going to handle. Here there are 4 biosafety levels are there. That's called BSL1, BSL2, BSL3 and BSL4. Among that, the BSL4, it is a high risk microorganisms, and BSL1, that is called the low risk microorganisms. This ranking of BSL1 biosafety law level 1, level 2, level 3, level 4, it is mainly based on research and based on what type of microbes they are going to handle. First, let us see about the biosafety level 1 and their procedures or principles or protocols that we are going to follow. This is biosafety level 1. It is not too much to be worried. This is the lowest biosafety level. And in this level, they are going to work on the microorganisms which are not pathogenic, which are not at all pathogenic. So what type of precautions they have to carry here? Always you should use mechanical pipetting instead of pipetting with the help of the mouth, and safe handling of the sharp objects. Splashing of aerosols or splashing of chemicals are not allowed, and frequent washing of hand need to be done in that particular place. And that particular place, laboratory, it should be free from drinking, smoking, or normal drinking of water and eating of food. All those things need to be should not be done in that particular place. And people, those are working there, they should have a proper gloves, and then they should have the goggles, lab coats, gowns, and all those things to protect themselves from that level of pathogens. And all the infectious material, what they are going to use it must be decontaminated before being disposed of. It should be properly cleaned. That is about the biosafety level 1. Next, we are going to talk about biosafety level 2 after 5 minutes. Just go through it, children. After 5 minutes, I'll explain to you. The next is about the biosafety level 2. Biosafety level 2. So let us learn about the biosafety level 2. The biosafety level 2, it is little more pathogenic organisms we are going to handle when compared to level 1. In this biosafety level 2, we are going to handle the organisms like organism which causes human diseases in human, like encephalitis, human immunodeficiency virus, and Staphylococcus aureus are some pathogenic organisms. Especially in this biosafety level 2, the researchers or the scientists, those who are working there, they should be take care of themselves properly. They should not have any injuries, any cut or injection should not be there. And what are the biosafety procedures they have to follow in the laboratory? That is, always they should wear goggles, glasses, face shields, mask, all those things. And when you are doing the procedure they have to do it in a closed biological safety cabinets, because they are handling the microorganisms that need to be taken care. And if there are waste materials produced in the laboratory, immediately it has to be contaminated. The decontaminants, immediately it has to be disposed. Before disposing, incineration need to be done. It has to be burnt so that it will not cause any infection to the surrounding in which it is thrown out. And also an eye wash and a sink must be there. If they are exposed some pathogen, immediately they have to wash their faces, eyes. For that, all the provisions need to be provided. And also the biohazard signs like this need to be sticked here and there to inform that they are in the biohazard areas. Next, safety level 3. In safety level 3, it is much more pathogenic microbes which causes serious disease when it is inhaled. Like example, the COVID virus. So here, those who are working here in the biosafety level 3, they need to be more precautious than any other levels. They should have the respirators like this, because those pathogens, it can even easily, it can be inhaled through the nasal openings. So before that, it has to be filtered. For that, they have to use the respirator. And also, if they are doing some experiment, it should be in a closed biosafety cabinets. And this research laboratory, it should be little bit away from the general building, but there should be an access from that. And all the researchers, those who are working in that laboratory, are to be under medical surveillance, and they are to be immunized against certain microbes. If they are exposed to, if they are going to work on the new microbes, before that they should take vaccination to protect themselves from that particular pathogens. All these things need to be done. Next is biosafety level 4. This is, in this laboratory, they are going to handle very high dangerous as well as exotic microbes, that is from the foreign countries. And handling this microbes or improper handling of these microbes can lead to death of a person, like Ebola, Marburg virus, like that. If they are going to handle, that comes under the biosafety level 4. In this level, what are the precautions, the practices they have to follow? The researchers should change their clothes and shower while exiting. They should have a proper covering like this. When they are entering the lab, they have to be fully covered like this. When they are exiting, they need to take a shower while exiting with proper cleaned water or some alcohols. And all materials should be decontaminated. All the equipments what they have used there, it should be decontaminated. And also, again, when they are doing the research, everything should be done in a safety cabinets. And this laboratory alone, it should be away from the general building, and it should be a separate building, and entry into that zone, it has to be completely restricted. Only the authorities, only the person who can handle those disasters, or those who can handle that particular viruses, those who know the safety rules properly, those people alone, it will be allowed inside. Other people, they are not allowed because the restricted area is a biohazard area. These are the safety rules are there. Next topic what we are going to learn today, it is about biopiracy. Biopiracy. So what is this biopiracy is all about? Biopiracy means any illegal operation of living organisms, microorganisms, plants and animals, and their traditional knowledge, are illegal use of microorganisms, plants or animals without proper authority, are using an indigenous knowledge of a particular country or a particular community or a particular people, without providing any benefit to them. If you are going to use their knowledge, that is called as biopiracy. If those knowledge are organisms or plants or animals, they are used by others' profit without the permission of the particular concerned people or concerned authority, without any compensation or without any proper permission from that people, that is called as biopiracy. So this biopiracy, there are various threats posed by biopiracy. There are certain knowledge or genetical resources that are belonging to a particular region or particular community or a particular country or a particular state. If it is stolen or claimed as one's own, an individual could have been identified a new thing, whereas some other company can take ownership over that. Use of a knowledge or genetic resource in that area of its origin or the traditional usage, if you are going to hamper that, that is the threat to biodiversity. If a patent holder for a particular product, if unfairly he get profit from the patent, that is also a threat for biopiracy. And if a patent claimed and awarded illegally and unethically somewhere in the world, that is also a big threat for the biopiracy. Now let us see about an examples of biopiracy. So the fruit what we are seeing it here, this is called as African super sweet berries. This is Pentadiplandra brazilin. This is a particular fruit, a plant that is found in South Africa, west region of the South Africa, and this is a very important source of protein. It has a protein, that protein is called brazin. People use this particular brezin because it is a low calorie sweetener and it is 1000 times sweeter than the sugar. Recent developments, including isolation of the gene responsible for sweet berries, brezilin, it has been sequenced and patented in the USA. But which country it belongs to? Africa. This is called biopiracy. Next example, patenting of Asadicata indica, neem. Neem, this particular species, it is belong to our own land, India. Indians have shared the knowledge about the neems, regarding neems throughout the globe. But in the year 1994, the US Department of Agriculture and an American company that is called W.R. Grace, they have received an European patent to treat the neem oil for fungal infections. This is also an example of biopiracy. Next is the biopiracy of enola bean. This enola bean, it is a bean variety that is found in Mexico, the North Mexico. This enola bean, it was named after a person called Laurie Proctor, who patented in this particular bean in the year 1999. The farmers in the northern Mexico, they actually depend on this particular bean. They depend on the sales of this particular bean, production and sales of the bean. Whereas this patent holder, the Larry Proctor, he sued a large number of importers of Mexican yellow beans. He got lot of money, but it causes lot of economical problems or economical damage to the North Mexican farmers. So the North Mexican farmers, they filed a suit against that particular person, and they revert back the patent that was issued by the US Patent and Trademark Office, and finally the farmers, they got the favor of using that enola bean, where the patent was provoked. Next is the rosy periwinkle. This is a scientific name of rosy periwinkle. It is called Catranthus roseus, which was mainly found in Madagascar, but nowadays this particular rosy periwinkle, it is present throughout the world. Throughout the various tropical countries across the globe, we can see this type of flowers, this type of plants, and this implements the researchers can obtain knowledge from one nation and plant samples in another nation, they can able to share it, but the original species, it belongs to Madagascar. Next example is Haldi patent. Curcuma longa. This is a particular herb, a tropical herb grown in the eastern part of India, and it is called as the rhizome. In the year 1993, a patent was filed by University of Mississippi, the medical center, and also the applicant received US patent for using this turmeric powder to treat the wound healing patent. But in the year 1997, the Indian government, that is the Council of Scientific Industrial Research, objected the patent issued by the US patent, and they filed that the turmeric patent failed to satisfy the criteria of novelty, because the ancient medical textbook which was found in India or which was published in India, they have more knowledge about the Haldi. when compared to the patent holder. So the patent was cancelled in the year 1997 and we have got the patent again. Similarly, the basmati patent. Basmati rice, it is an exclusively grown in subcontinent, the Indian subcontinent. Along with that, it can be seen present in like Pakistan, Bangladesh, and so on. The Indian government, they had a argument with the US protocol, PTO. They've granted patent for using basmati rice. They've given the rights to a US company which is called Rice Tech. This Rice Tech company, they've got patent for this particular rice, usage of the basmati rice. And Indian government, they filed a case against that, and they have revoked the patent. And now the patent was given to both the countries, where in US they are using this particular basmati rice in the name of Texmati, whereas in India we are using that as basmati. This is how some of the examples of biopiracy. So with that, we'll come to the end of this particular lesson, that is the biotechnology and its applications. Children, so far we have completed 2 complete chapters. One is genetics and the biotechnology, applications of biotechnology. Next topic what we are going to learn today for another half an hour, it is going to be the human health and diseases, biology and human welfare. First I am going to explain to you about human health and diseases. That is the topic what we got to learn today, and we'll see it after a break. We'll go for a break for 5 minutes, and after the break I'll come and explain to you about, we'll start the new topic. Dear children, we are going to start a new topic today. The topic is human health and diseases. Human health and diseases. So let me define what is a disease. A disease, it is an abnormal condition affecting a living organism. We can define a disease when a part of a body, if it is not functioning properly or if it is disturbed by certain means, that is called as a disease. Usually a disease, we can exactly tell it is a condition where a person is infected by a pathogen or due to certain improper functioning